Hello there, my name is Anish Kirtane, a PhD student at ETH Zurich, and in this presentation, I want to ponder the question, are lakes accumulating global biodiversity information in the form of environmental DNA, or eDNA as we like to call it? Can we go out into a lake, collect a few water samples, and find out all the organisms that have been living in the watershed and shedding their DNA? Let's get started with the problem at hand. And I don't think I need to explain the problem or convince anybody, especially at this conference, but we have about 1 million species at risk of extinction on this planet. And that is just the species that we do have a good idea about, mostly vertebrates. And it's a result of the fact that how difficult biodiversity monitoring is. Landscapes are huge. Organisms, especially the ones that we're interested in, tend to be quite rare and hard to find. Some require a lot of taxonomic expertise. It's expensive. So we ought to look for new technologies and creative methods for evaluating and monitoring biodiversity. How do we protect and conserve something if we don't know they're present or absent? So let's get to environmental DNA, or as I said, eDNA. Uh, so eDNA is DNA. So all forms of life are based on DNA, and all organisms release their DNA into the environment. It could either be through dead cells, clumps of tissues, through feces. And this DNA, even though if it's shed on land, uh, through rainfall can get transported into waterways and eventually into lakes where we can collect this DNA. A key parameter, the key concept that actually makes eDNA useful for monitoring current biodiversity, the current occupancy, is that that DNA degrades relatively rapidly. So DNA, of course, it's complex. It can be in many different states. DNA can be bound inside a cell. It can be free-floating DNA. It can be inside organelles such as mitochondria, or it can be bound to particles. And all of these things determine its degradation rate. There are also environmental factors such as temperature, pH, bacterial activity. So UV light degrades DNA. Enzymes require a certain temperature and a certain pH to work. DNA is full of nitrogen and phosphorus. So naturally, bacteria want to uptake and break it down. So DNA in environment persists at different time scales, but most of the experimental evidence shows that DNA persists between hours to days to maybe weeks, again, depending on environmental conditions. So we can get a relatively good idea of the organisms living there if we find traces of their environmental DNA. And at this point, I must say that the kind of information we get from environmental DNA data is not exactly the same as conventional methods. We might not be able to pinpoint exact location to where the organism is, or be able to tell the exact number or biomass of the target species. What we get is a broad resolution understanding of who all is living in the watershed. And of course, we can do additional sampling to source track where the DNA is coming from. But essentially, it's a broad scale tool that can help us not look for just one or two target species, but almost all life living in the landscape. For instance, I really want to highlight this one study done by my advisor, Christy Diner, where she really evaluated the idea that rivers are acting as conveyor belts of biodiversity information. She found that river water contained not only a few forms of life, but DNA from terrestrial life, from aquatic life, and from many different phyla, from many different diverse families. And this really opened up the doors to use this technology, environmental data technology, for monitoring uh, all forms of life, and terrestrial life and aquatic life. And this is what a lot of other studies have extrapolated off of and advanced the concept, and this is what we are using as well for a global biodiversity monitoring project. I'll get to that in a second. But first, I want to talk about transport of DNA, and this does relate to the degradation we spoke about earlier, but we can think of our landscapes as networks that have sets of links that transmit information and nodes that receive them. We can think of internet or neurons, but in our case, it's the waterways. The rivers, the streams act as links that transmit this biodiversity information to lakes. So if we are able to go to these lakes and collect some water samples and analyze the DNA, we might be able to find out everything that's living in the watershed. However, this is based on one key hypothesis, that the amount of time it takes for the transport of DNA should be more than the time it takes to degrade that DNA. And this figure is quite good at illustrating the same concept. There are organisms in the waterways as well as on the land, constantly releasing their DNA. Now, this DNA might take, let's say, a day and a half to reach, to get washed off and reach a lake. In that case, the DNA should not degrade faster than 1.5 days if for us to be able to confidently say that we have surveyed the biodiversity of the landscape with environmental DNA. And this especially will be important for our global project, where we have a grant to survey 120 lakes from around the world to really determine the reproducibility and reliability of these methods. Again, a lot of eDNA surveys have been done in small pockets. However, a global study like this has not been conducted yet. We are working with a collaborator in Canada in McGill University, Bernhard Lenner, who has mapped all lakes, and based on how much time it takes for the water molecule from the edge of the watershed to reach to the lake. And what we found out that most of the lakes in the world, as you can see, there are these tiny dots on the map. Each of the tiny dots refers to a watershed or a lake, and the size of the dot shows you the amount of time it takes for the water to reach the lake. Most of the lakes in the world are in between a few hours to a few days of transport time. And this, if you remember how quickly DNA degrades, it is between hours to weeks, depending, of course, on a lot of other environmental conditions and the chemical factors in factors in the water. So it turns out that most of the lakes in the world can be sampled and can be used for global biodiversity monitoring. And here's the interesting part. All these 120 lakes that we will sample will, of course, help us create this global biodiversity atlas. But we can also test a bunch of other hypotheses. We can use the biodiversity monitoring information to improve and further eDNA science. So one of our ideas, one of the sub-projects in the global sampling is to sample along the migratory pathways of birds, especially birds that fly in large numbers. In the global project, we are going to be taking water samples through many different time points, in which case we will detect migratory birds coming in, and then we will also see them leave. Migratory birds are also really well tracked. All the ornithologists watching this will know that people are very enthusiastic to know how birds migrate, where they fly. The tracking is done really well. So this can be a natural degradation experiment of dozens of bird species at the same time as they move along our group of lakes that we are sampling. When the birds come in, we will see a rise in the DNA concentration of those species. And as birds leave a given lake, we might see a degradation, a lowering of eDNA concentration. So we can really evaluate at different lakes for how long does this bird eDNA persist. We can look at many different species at the same time. And this is just one of the hypotheses, one of the many hypotheses we can test with the kind of data we get from environmental DNA and sampling lakes from around the world. However, we cannot do this alone. Our sampling campaign, the global sampling campaign, will start in 2022 and go into 2023. We are looking for collaborators around the world, scientists who are interested in biodiversity monitoring, scientists who are interested in environmental DNA, and we really want to have a consortia of scientists from diverse geographical regions, and hopefully we can sample in many different countries around the world. So if you are watching this presentation and are interested in collaboration, feel free to contact me or my advisor, Christy Diner. Our email addresses and Twitter accounts are written here. Feel free to take a screenshot, write it down. And thank you so much for your virtual attention. I cannot wait for the live session where I can talk to some of you and answer all questions you have. See you then.